Hello, my name is Taylor Rains, and today we will be talking about point mutations and their effects on amino acids. Point mutations are mutations that result in the substitution of one base for another. There are two specific types of point mutations. The first of these two types is called a transition. This occurs when a purine changes to another purine or a pyrimidine changes to another pyrimidine. The second of these two types is called a transversion. This occurs when a purine changes to a pyrimidine or a pyrimidine changes to a purine. Point mutations can have 4 different effects on the amino acids that are coded. These are known as missense or non-synonymous, nonsense, silent or synonymous, and read-through. To discuss these effects, let's look at this example coding strand so we have something to mutate. Our first step is to find our normal template strand. This is when we match up A's to T's and G's to C's. The second step in this process is to then code for the mRNA. This is just basically going back to the strand we had before, but changing all the T's to U's. Now that we know our normal mRNA, we can now find out what amino acids this sequence codes for. As you can see, about halfway through the sequence, there's a stop codon, so we stop coding from here. So now that we have an example, let's see how the different point mutations can affect our mRNA sequence. Our first effect is known as missense. A missense changes the amino acid by changing one of the bases. In looking at our example coding strand, we can see how changing one base changes the entire amino acid that is coded. A neutral missense is the same thing, but the mutation has no effect on the protein function. The next effect is known as nonsense. This changes the codon from a normal amino acid to a stop codon. Again, by looking at our original coding strand, we can see by just changing one base, the guanine to an adenine, we change the entire codon into a stop codon. The next effect we will discuss is the silent effect. This is when a base is changed, but the amino acid that is coded is the same. From looking at our example original strand, we can see by just changing ACC to ACT, nothing changes with the amino acid. The last effect we will discuss is read-through. A read-through is basically the opposite of nonsense. A stop codon changes to an amino acid, and this causes a longer protein to be coded. If you remember, our original coding strand had a stop codon almost in the middle, at TGA coding for UGA. A read-through mutation at this location would allow for us to code the rest of the remaining amino acids. To accomplish this, we could mutate the TGA into TGG. This would cause the template strand to then be ACC instead of ACT, which would then code for the mRNA sequence of UGG. This, instead of being a stop codon, would then code for an amino acid. So now that we have a new mutant, instead of stopping in the middle of the coding sequence, we would now continue coding and have a longer amino acid chain.